All right. So this is the Moodle site. Hopefully you've all gone and looked at it. I will generally organize this by date. So for example, there's a folder here called September 3rd Materials. Um, it's got the PowerPoint that we're going to be working on today. It also has the Plants and Society chapter. Plants and Society is this textbook that is intended for non-science majors who are taking a course that would be called something like Plants and Society. It's not necessarily a textbook intended for biology majors, although it does a pretty good job of covering the science. And it usually runs about 80 bucks, and so I've opted to not have you buy it because we don't use a significant amount of it over the course of this semester. So um, there are 3 chapters here at the beginning in that book that are really useful for understanding some of the basic plant biology that you need to know to understand the first part of Paulin's book. So we'll be reading some chapters out of that. In this, we've also got the food blogging prompts are here. So what you do for week 1, what do you do for week 2, etc. And actually, these dates are wrong because these dates are last year's dates, I think. So I need to modify that. But these are due at the end of this week, i.e. today, and then at the end of next week. But once again, as I've said before, space them out over the course of the week so that you're not just getting a bunch of posts being made Thursday night at 2 in the morning. Um, so that's that. Um, Questions about what we were talking about last time? Did you go and read chapter 1 of Poland between last time and this time? Did me kind of going through what I wanted you to kind of be paying attention to help you as you were going through that? Okay, good. Um, so in Poland's book, he has this section of the chapter that he has titled Corn Walking. And at the beginning of this section, he writes, descendants of the Maya living in Mexico still sometimes refer to themselves as the corn people. The phrase is not intended as a metaphor, rather it is meant to acknowledge their abiding dependence on this miraculous grass, the staple of their diet for almost 9,000 years. 40% of the calories of a Mexican eats in a day comes directly from corn, most of it in the form of tortillas. So when a Mexican says, I am maize or corn walking, it is simply a statement of fact. The very substance of the Mexican's body is to a considerable extent a manifestation of this plant. So there's truth to this idea that you are what you eat. And he goes on in this chapter to talk about kind of how corn is ubiquitous in our food system. If you're drinking a soda, you're drinking a soda that has a lot of high fructose corn syrup, a type of sugar that is derived from corn. And because we have such an abundance of cheap corn, which we'll talk about later in this semester, corn has been repurposed into all sorts of other things. It's in the adhesive that holds the paper on the drywall in this room. It's in the ceramics that are in spark plugs. It's actually used to make the Coke bottle that the high fructose corn syrup inside of the Coke is holding. So it's been repurposed into plastics and all kinds of goods, simply because we grow a lot of it. Because we grow a lot of it, it's relatively cheap. And whenever you have a cheap resource, people tend to find purposes for it. Purposes beyond just simple food. You just raised your eyebrows. Why are you surprised at that? No, in the back. What's your name again?
Oh, Aiden.
Hmm?
I didn't mean
Aiden.
to.
Oh, yeah, you were like, whoa.
Oh,
Oh,
sorry, no, I was probably just like waking up my eyes.
okay, all right. Are you guys familiar with a economic principle called Say's Law? Anyone? Business people? So you know about the law of supply and demand. As the supply of a source of a resource increases, that resource becomes cheaper over time in the face of a constant demand. If demand rises and supply doesn't adjust, then price rises because the supply isn't satisfying the demand. You have a resource that is in shorter supply. This has happened to lumber, for example, in the last year, if any of you have been using lumber for anything. But Say's law basically says supply creates its own demand. And what this is basically saying is that if something is in high supply, it's relatively cheap, and then because it's relatively cheap, people will find uses for it because it's a cheap commodity to acquire. And this is sort of what has happened with corn. We've repurposed corn to all these other purposes, make all these other products from it, mainly because we have a high supply of it, and as a result, it's very cheap. And so he talks a little bit about this on the first paragraph on chapter 20. But then he starts talking about carbon. And so on page 20 and 21, he starts talking about carbon, and he starts talking about the differences between C3 and C4 plants. C3 is one photosynthetic pathway. It's one way in which plants can make sugar out of carbon dioxide. There is another way called the C4 photosynthetic pathway. Now, there are some fundamental differences between these two photosynthetic pathways. C3 plants are things like wheat is a C3 grass, soybeans are a C3 legume. Corn is a C4 grass. So within our food supply, we have these two different kinds of plants that do two different kinds of photosynthesis. Ultimately, C3 pathways are found in plants that do better in cooler, moister environments, and these plants tend to do better in hotter, drier environments. And the reason that these plants thrive in these different environments has everything to do with the way in which they go about doing photosynthesis. The other thing that happens, though, is that these two photosynthetic pathways process carbon isotopes differently. So in both cases, they process carbon isotopes differently. So when you get over to page 22, so starting on page 21, it says, at its most basic, the story on life is the story of life on Earth is the competition among species to capture and store as much energy as possible, either directly from the sun in the case of plants or in the case of animals by eating plants and plant eaters. The energy is stored in the form of carbon molecules and measured in calories. The calories we eat, whether in an ear of corn or a steak, represent packets of energy once captured by a plant. The C4 trick helps explain corn's plant success in this competition. Few plants can manufacture quite as much organic matter and calories from the same quantities of sunlight and water and basic elements as corn. 97% of what a corn plant is comes from the air, 3% is from the ground. The trick doesn't yet, however, explain how a scientist could tell that a given carbon atom in a human bone owes its presence there to a photosynthetic event that occurred in the leaf of one kind of plant and not another. In corn, say, instead of lettuce or wheat. The scientist can do this because all carbon is not created equal. Some carbon atoms, called isotopes, have more than the usual component of 6 protons and 6 neutrons, giving them a slightly different atomic weight. C13, for example, has 6 protons and 7 neutrons, hence C13. For whatever reason, when a C4 plant goes scavenging for its 4 packs of carbon, It takes in more carbon-13 than ordinary C3 plant, which exhibited a marked preference for the more common carbon-12. Greedy for carbon, C4 plants can't afford to discriminate among isotopes and so end up with relatively more carbon-13. The higher the ratio of carbon-13 to carbon-12 in a person's flesh, the more corn has been in his diet or in the diet of the animals he or she ate. As far as we're concerned, it makes little difference whether we consume relatively more or less carbon-13. It just gives you this signal that you're eating more C4 plants. And we actually get double doses of C4 plants because another C4 plant that I should have mentioned a few minutes ago is sugarcane. So if we have a diet that is high in corn-based foods or high in cane sugar-based foods or high in both of those things, we're going to have a higher C4 representation in the atoms in our bodies. So here's this first little bit of science that comes at you in chapter 1. We're at page 22 of the book as a whole, and we're faced with this idea of isotopes. Did you guys understand what he means when he talks about isotopes? No. Okay. So, you guys have all this? I'm about to erase it. Okay. So on page 20, he talks about stable isotope analysis, talking about taking a sample of your fingernail, running it through an isotope analyzer, and it will tell you how much carbon-13, how much carbon-14 you have by analyzing the actual carbon atoms that you have. So that's how you know, that's how you can tell if a person is consuming a large amount of C4 plants, is by looking at the ratio of these isotopes. So essentially, to understand page 20, 21, and 22, you have to understand stable isotope analysis. This is just a mechanism. We use it in ecology a lot to examine the diets of organisms. So for example, there are different isotopes that occur in the oceans rather than occur on land. And in Puerto Rico, where I've worked before, there are these shrimp that live part of their lifespan up in terrestrial streams, and they spend part of their life down in the oceans. And when they come back up into the headwaters of these mountain streams, they bring all of those marine isotopes with them. As their bodies die, insects in the streams decompose those bodies, and when those insects emerge from the stream, they fly out into the forest. And so they take those marine isotopes into the forest with them, and then when lizards and frogs eat those insects, we can pick up that isotopic signature in the frogs and lizards. And so among the things that I've done in Puerto Rico is looking at how export of energy and nutrients from streams into the terrestrial environment is influenced by what happens down in the estuaries in the oceans. You get this movement of nutrients up the streams, and then it gets exported out of the streams into the forest through these insects. And we do that all by recognizing that the ratio of isotopes in marine environments is different from the ratio of isotopes in terrestrial environments. So you can actually measure the weight of atoms, and you can figure out how much of the body of an organism is actually made up of C13, C14, or C12. And so this relies on knowing that atoms are made up of neutrons, protons, and electrons. These things are big. These are big subatomic particles, and electrons are tiny. They have negligible mass, very little mass at all. So most of the weight of an atom is in the protons and neutrons. Isotopes are versions of a particular atom that vary in the number of neutrons that they have. So normally when we think of carbon, for example, we think of carbon as having 6 protons and 6 neutrons. Oops. This then is carbon-12, so its kind of atomic mass, because a mass unit in chemistry is basically about 1. So 1 mass unit is about the weight of a neutron, 1 mass unit is about the weight of a proton. And so if you have 6 protons and 6 neutrons, you have a mass of 12. But you can also have forms of carbon where you have 6 protons and 7 neutrons. And you also have carbon where you have 6 protons and 8 neutrons. This then is carbon-13, and this is carbon-14. And so with the addition of each of these neutrons, that carbon weighs a little more than if you didn't have that extra neutron around. These are the 3 isotopes of carbon. If you go to a periodic table of the elements, let's find one that's relatively legible. Blow it up, and it's very blurry. You see that carbon is 6 in the periodic table, and that's because it normally has 6 neutrons in its nucleus. But you'll see here that the atomic mass of carbon is 12.001. Sorry, 12.011. You'll see that oxygen, for example, has 8 neutrons, so you would expect it to have 8 protons. So you would expect it to have an atomic mass of 16, but oxygen has an atomic mass of 15.996, I think. Is that right? Nope, 999. I'm off a little. So this is a little above 12. This is a little below 12. Well, when you see the atomic mass reported in a periodic table, it's the weighted average of the different isotopes of that atom. So in carbon, you have a lot of carbon-12, but you have a little bit of carbon-13, and you have even less carbon-14. But because you have some carbon-13 and carbon-14, that bumps the average of carbon's atomic mass up a little bit because it's the weighted average of all the different isotopes of carbon. Same thing with oxygen. It's pretty close to 16, but there's a little bit of oxygen 15 floating around there. And so that pulls the average down a little bit, although not nearly as much as carbon is pulled up, because there are isotopes of oxygen as well. When you look at hydrogen, you see that its mass is 1. And that's because a lot of hydrogen exists just as a bare proton with no neutron at all. Because it has 1 neutron, you should expect it to also have 1 proton. But the fact of the matter is, its atomic mass is down close to 1 because a lot of hydrogen just floats around as a proton. So these are actually heavier, even though the difference in weight is really, really small, but it's detectable. We have instrumentation that allows us to do this. We didn't have this instrumentation 50 years ago, but we have this instrumentation now. So through this, if you eat more C4 plants, you tend to have more of the heavy isotopes. So in the book, Paulin says, for whatever reason, when a C4 plant goes scavenging for its 4 packs of carbon, it takes in more carbon-13 than ordinary C3 plants do. And so he puts this out there, for whatever reason. Well, the fact of the matter is, there is a reason. The fact that Paulin doesn't know enough about photosynthesis of plants to write about this in the book doesn't mean that we don't know why this is the case. It's just that he either doesn't know it or thought that it was too much detail to go into to put into a book written for the general public to read. It's not a mystery. We know why C4 plants fix more heavy isotopes of carbon than C3 plants do. So this for whatever reason is knowable. Just because it's a question in the book doesn't mean that it's not knowable. Actually, it doesn't mean that we don't know it. So in order to understand what that reason is, you guys need to know something about photosynthesis. In order for you to know something about photosynthesis that is important from the relevance of why Mayans and Americans look different from Europeans when we look at your isotope ratios, you need to know about the difference between C3 plants and C4 plants. And in order to understand those differences, you need to understand some things about plant anatomy, really. Plant anatomy. So today what we're going to start doing, this is just to get you set for what's coming ahead. Today what we're going to start doing is we're going to start talking about plant anatomy. I'm not going to tell you everything there is to know about plant anatomy. There are some things that I'm going to mention, and when I mention them, I'm going to say, this is all you need to know about it. I might tell you a little bit more about it just for clarification, but I'll go back and say, but you don't have to know that for a test or anything. You just need to know this for a test. I'm only giving you enough plant anatomy to get us to the point where we can talk about photosynthesis effectively. And I'm only going to give you enough about photosynthesis so that you can understand what these reasons are that seem mysterious to Michael Pollan about why we can tell the difference between people who eat a lot of corn in their diets relative to people who don't eat a lot of corn in their diets. So that's the journey that we're about to begin with. Questions about that? All right. If you haven't read Chapter 3 of Plants and Society, definitely do that before Monday because this is the information that is in that chapter. So, you need to be able to understand the basic structures of plant cells and plant bodies in general, and you need to understand the difference between dicots and monocots. So dicots, once again, I will always be relating these things back to real organisms. Dicots are things like spinach, cabbage, any of your kind of leafy greens that you think about in the store, beans, soybeans. Monocots are, for the most part, things like grasses. So in agriculture, we would consider this to be wheat, corn, sugarcane, etc. These are all monocots. And monocots and dicots have fundamentally different structures to them, and so knowing the difference between those becomes important because they both make up our food supply to a certain degree. For understanding photosynthesis, you really need to understand the structure of the inside of a leaf of a plant. And so we're going to actually go into quite a bit of detail about the leaf structure because it's important for you to understand how that leaf structure relates to photosynthesis, how that leaf structure relates to the storage of the products of photosynthesis, the carbohydrates that photosynthesis makes, and then be able to explain these in terms of transport of nutrients and fluids throughout the plant body, and knowing about the roots, which absorb nutrients that they need to survive, and once again, pulling all of this back to photosynthesis. So that's kind of what the goal is for you to have a good working knowledge of just kind of how plants are structured. So plant cells are different from animal cells in a number of different ways. Plant cells usually have in the center of the plant cell, there is a very large thing called a vacuole. It's just a big membrane-bound sac, and it's filled mainly with water, but it's also filled with all sorts of solutes. So it will have sugars, starches, and things like that inside of it. Around the central vacuole is where all of the cell contents that have to do with functioning of the cell are. And the main ones that you need to know about at this point you have these things called chloroplasts. Chloroplasts are little organelles. So chloroplast, it's an organelle. It's kind of a teeny tiny organ, but it's the organelle where photosynthesis actually occurs. So that's the site of photosynthesis. We're going to talk about the nucleus later on when we start talking about genetics, but we'll only talk about it just briefly. So I'm not going to go into it now, but this is it right here in purple. And then the other thing that is useful to know about plant cells is that around the cell has secreted cellulose to create this kind of semi-rigid box that goes around the cell. And so this semi-rigid box is the cell wall. And how many of you have ever had a house plant that you didn't water? Okay. When you don't water your plant, what happens is the fluid that is inside the central vacuole diffuses out of the cell. And when it does, that cell is no longer putting pressure against the cell wall, and the cell wall becomes looser, kind of like when you lose air out of a tire. The thing that makes your tire firm is when you have a bunch of pressure inside the tire pushing out on this semi-rigid structure, the tire itself, that doesn't want to give. So you put more pressure in, the more pressure you put in, the tighter that thing gets. When you let the air out, that thing begins to deflate. That's with air inside of a tire, but the same thing happens with water inside of a plant cell. Water flowing into the plant cell pushes out on the cell wall and makes that thing more rigid because there's pressure being exerted out on this thing that doesn't want to be pressured. And so when your plant starts to wilt, that's the beginning signs that you haven't watered your plant enough because it's losing that pressure that pushes out on that cell wall. And so cell walls help plants maintain their structure. We maintain our structure because we have an internal skeleton. Plants don't have that. All of their structure is supported by the semi-rigid cellulose cell walls that go around their cells, and this turgor pressure, this pressure that I'm talking about, is called turgor pressure. And that's what keeps your spinach nice and firm and keeps it from falling over, is this pressure exerted by water. This is a schematic diagram. This is what it looks like when you look at it under an electron microscope. But basically, you have all the same components. You have the central vacuole. You have the cytoplasm out here. There's a nucleus here. And there's this rigid cell wall. And then here are chloroplasts. And inside of these chloroplasts are starch grains. So chloroplasts create sugar from carbon dioxide that's in the air, and then that sugar gets stored in long-term form in the form of starch. And so it's pretty common to see starch grains inside of the chloroplasts. Because they have this rigid cell wall around them, they have to communicate from one cell to another, and they do it through these pores that go from one cell to another cell through the cell wall. And so these things are called plasmodesma. You don't need to know that. It's just on there for the purposes of completeness. But you do need to know that even though there's a wall around each of these cells, they're not totally isolated from one another. They actually have these connections in the form of these little tunnels that go through the cell wall, connecting cells to one another. This is important because plants have to transport materials. Plants absorb water and nutrients in the roots, and they send that water and nutrients to the leaves at the top of the plant. And the way that they do that is by transferring materials from cell to cell through these little tubes, these little tunnels that connect cell to cell. And so there are these things called vascular tissues. Let me write that on the board. and they transport materials around the plant in the same way that your vascular system transports materials around your body. You have a heart and arteries and veins that transfer fluids around your body. Those fluids also contain things like red blood cells and stuff like that. Plants also have a vascular system, and that vascular system is facilitated by these tunnels that connect cells to one another going through the cell wall. So just in general structure of a plant, their root is the part that is below the ground of a plant. The shoot is the part that is above ground of the plant. And so this ground-atmosphere interface is kind of the dividing line between where the roots are and where the shoots are. And once again, you have these vascular tissues that take fluids from the roots, and this vascular tissue is connected from the root to the shoot and then goes out actually into the leaves in the form of these veins. And there's two types of vascular tissue. One vascular tissue takes stuff from the root to the shoot. Another vascular tissue takes stuff from the shoot to the root. So one is an up elevator and one is a down elevator for the transport of materials. The other thing you need to know about is meristems. Meristems in plants are the growth tissues of plants. So when a plant grows, it can grow up, can get taller and taller and taller, or its roots can grow deeper and deeper and deeper. But the other thing that it can do is it can grow in girth, in how big the stem is. And so these meristems are the only places in the plant body where growth occurs, where cell division is actively happening. When you grow all over. Your muscles grow, your skin cells grow. There are all kinds of places in your body where you grow. So your cells have the ability to go through the process of cell division all over your body. Plant cells only have discrete little sections of their bodies where growth happens, where cell division happens, and it's in these meristems. And these meristems occur in three places. There is a meristem at the tip of the root that allows the root to elongate, to get longer. There is court cambium in the root that allows the root to get thicker around. There are apical meristems at the tips of the shoots that allow plants to elongate. And then there is a vascular cambium here around the edge of the plant that allow the plant to get bigger in diameter. So the roots and the shoots are kind of similar, and that there are areas of cell division that allow the plant to get longer, and there are areas of cell division that allow the plant to get thicker. And we just call them different things depending on whether they're in the shoot or whether they're in the root. These meristems can be located in different places in plants, and that's one of the fundamental differences between monocots and dicots. So just store that away for a minute. We'll get to that later. There are also what we call lateral meristems that are located at these buds where a new leaf buds off, and that allows for the growth of new branches. And normally, these things don't put out new branches unless something happens to the apical meristem. Do any of your parents have hedges? Nobody? You got a hedge? Do you ever have to trim the hedge? Why do you trim the hedge? Yeah, so if you have a hedge, and it's all nice and trimmed, a bunch of little stems that is the main trunk, but then the hedge is trimmed because are you good at trimming the hedge? Yeah, you're all right at trimming the hedge. So if you leave it for a while, what starts to happen to the hedge? It doesn't get big, it gets straggly, doesn't it? Because what happens is you start to get branches growing up. Well, these branches growing up are because this apical meristem is still there, and it tends to continue growing up. As that apical meristem grows up, it produces a hormone that tells these other meristems, these lateral meristems in the buds, it tells them to not undergo cell division. So it keeps them in bud form. When you come through and you trim that hedge, you get rid of all those apical meristems. When you get rid of those apical meristems, the hormones that are being produced that suppress growth in these lateral buds goes away, and all of a sudden those lateral buds are able to grow. And when they grow, they put out little lateral branches. Eventually, if you neglect your hedge, those lateral branches get so tall that they start poking up out of the top of your hedge, and then your parents harp on you to go out and trim the hedge again. So we trim our hedge for two reasons. One reason is because our neighbors will complain if our hedge looks like this. You might get calls from the city saying, hey, your hedges are getting a little sloppy, you need to do something about that. But the other reason is that whenever you trim your hedge, it cuts off that hormone supply and it makes all these little lateral branches grow. Why do you have a hedge? Do you typically have a hedge in the center of your front yard? No, where do you have hedges? Around the edges of your front yard. Why? Yeah, it's kind of a barrier. You have a hedge to keep neighbors out of your business, don't you? Well, as you trim that hedge and these lateral meristems grow, your hedge gets thicker. It doesn't get taller, it gets denser inside of there. And that's what you want a hedge to do. It's kind of a privacy screen. Well, a privacy screen can be really, really thin so that you can see through it, or it can be really thick and densely packed so that you can't. And so one of the other reasons that you trim your hedge isn't just to keep it tidy, but also it creates a growth in the inside of the hedge because it frees up those lateral meristems to grow because you've gotten rid of the apical meristems. Now you'll never view hedge trimming the same again, hopefully. So this apical meristem is designed to increase length. You cut that off, that frees up these buds to form new branches, making the plant bushier. You guys grow tomatoes? I like tomatoes. We have a ton of tomatoes right now. We've grown our tomatoes this year in raised beds with trellises supporting the weight of the plant. And because we grew them in trellises rather than like in a basket that kind of holds and supports the whole plant, we've actually been cutting off lateral branches and allowing the plant to grow up, relying on the trellis to support it. If we were growing them in one of these cone-like baskets, we would be trimming the apical meristems to get it bushier to fill up that basket. And so depending on kind of what your agricultural strategy is and how you're growing a particular crop, you may trim the apical meristems off to promote bushiness, or you may trim the lateral stems off in order to encourage height. And it just depends on kind of what your goal is and how you're growing something. So once again, knowing something about basic plant biology influences, for example, in my case, how I'm growing tomatoes differently this year than how I grew them last year. The bad thing about that is I built a 7-foot trellis for my tomatoes, and my tomatoes are now like 12 feet tall. So I need to balance height versus bushiness next year, perhaps. And I have more tomatoes now than I can possibly eat. I'm constantly giving neighbors tomatoes. So these axillary buds are controlled by the apical meristem, and so that makes the plant more bushy. This is actually a figure from the textbook, if I recall correctly, and it just kind of organizes the tissues. You're not going to be tested on this, but it's basically just a schematic diagram of the things that have been in this last couple of slides relative to tissues. We have apical meristems and lateral meristems, and then it tells you what the different kinds of tissues are. Really, you don't need to know much about it beyond this and that. This stuff in the middle you don't really need to know, but we are going to talk about primary growth versus secondary growth at some point. So there are these tissues in plants just like you have tissues in you. The two most important tissues that we need to think about are xylem and phloem. Xylem is the tissue that transports fluids from the root to the shoot. And that's really all you need to know. It's like a hollow tube that takes fluids from the root up to the shoot. There's some other detail on there, but if you remember anything about xylem, remember the direction of flow. It's the up elevator for liquid and nutrients. Phloem is the opposite. It takes stuff from the shoot to the root. So whatever products are being produced as a result of photosynthesis up in the leaves, those things are stored in the roots, and they get to the roots by traveling through phloem. So you have an up elevator and you have a down elevator. The up elevator carries water and nutrients. The down elevator carries water and the products of photosynthesis for storage in the roots. These two tissues are organized relative to one another. And so this is what a monocot looks like. This is what a dicot looks like when you look at the stem of the plant. In the monocots, these vascular bundles, these bundles of xylem and phloem, look kind of like a clown face in the monocots. And you see that the phloem is on the outside. That's taking materials from photosynthetic producing leaves down to the roots, and the xylem is on the inside. You'll also see that these monocots have these cells called bundle sheath cells. Bundle sheath cells are crucially important to know. These are cells that go around the vascular bundle. You see that in this slide they stain red, and they stain red because they possess lignin. So a vascular bundle in this case is made up of phloem and xylem, and they're surrounded by a vascular bundle. In dicots, you have phloem and xylem, and then there's this thing called the vascular cambium in between them. And the vascular cambium is one type of meristem. And the vascular cambium, what it does is it produces new phloem to one side and new xylem to the other side. And so this is how the plant gets thicker in diameter over time, is by producing xylem and phloem to either side of the cambium. You'll notice back on the slide, these vascular bundles are scattered kind of randomly throughout the dicot, but you can see that these vascular bundles are arranged in a ring around a dicot. The vascular cambium is in here. It's producing phloem to one side. It's producing xylem to the other side. So as it grows, it's just getting bigger and bigger and bigger and bigger. And this is why plants that are monocots, things like grasses, your grass gets taller and taller and taller and taller, but as it gets taller and taller and taller, it doesn't really increase that much in girth, in the thickness of the stem, because of the way its vascular bundles are arranged and the fact that they don't have this organized vascular cambium in the middle of all of that. So when you get trees, you've seen a tree that's been cut and you've seen the growth rings. Those growth rings are due to different rates at which that vascular cambium is producing new cells. During the growing season, when cell division is really fast, it's growing very quickly, and so it's producing a lot of phloem to the outside and xylem to the inside. During the winter, when the tree slows down in terms of its activity, it's producing less phloem to the outside and xylem to the inside. And so you get areas of growth that appear lighter typically, which is in the growing season, and then areas of growth that appear darker in a cross section of a stem that is due to winter growth. And it's all just due to changes in the rate of cell division. But as a tree grows, as a woody plant grows, produces more and more xylem to the inside, but as it produces phloem, the xylem is pushing out on that phloem and compressing it. And so the layers of phloem get compressed on one another over time, while the xylem just continues to make up the bulk of the branch of a tree or something like this. And so that bark that you see going around that's all this compressed phloem that ends up in the form of cork, and then you have living phloem here. In the old days, when you would go to harvest a forest, it turns out cutting green live wood is much more difficult than cutting dead wood, as it turns out. It's more resilient, it tends to bounce back when you shove an axe into it. If you've ever chopped wood, hacking on a thick log that is green, when you hit it with your axe, even if you have a sharp axe, it tends to want to spring back at you, whereas dry wood, your axe kind of sinks into it. And so in the old days, before we had modern methods of harvesting timber, lumber companies would send guys out into a forest, and they were equipped with nothing more than their camping material, a gun to hunt with, and an axe. And they would go and they would find trees that were of the suitable size that the lumber company wanted to harvest. And they would do this a couple of years before the lumber company actually went out to the forest. And what they would do is they would ring a tree. And what they would do is they would chop around the tree in a band. If this is your tree trunk, they would chop a band, chop around the trunk at one spot, chop around the band at another spot, then they would chop vertically, and then they would peel the bark off of that. And what they're doing is they're removing all of this material, all the way down to the vascular cambium that separates phloem from xylem. Now, at that point, what the plant can do is the tree can take materials from the roots and send them up to the leaves. And those leaves will do photosynthesis, but then when those leaves go to bring those products of photosynthesis down to the roots to store, they can't do that because that transport tissue has been stripped off by these lumberjacks. A couple of years of doing that, and now the tree doesn't have any reserves in its roots to support growth, and the tree dies. And then the next year, when the lumber company comes through, they see these trees that have been ringed, they see that they're dead, the wood has started to dry out, and they're much easier to harvest at that point. So, just an illustration to help you remember what xylem and phloem are doing. This takes stuff from the roots up to the leaves. This takes stuff from the leaves down to the roots, and that's a quick and easy way, cheap way to kill a tree is by getting rid of that transport mechanism. And it was used commonly, once again, back in the days, because it was just more time efficient to send two guys through a forest to kill a bunch of trees. It then made the harvesting of the trees easier when the time came. I feel like I've given you a ton of information. What questions do you have at this point? Jameson's like, oh my God, so much information. All right. So monocots and dicots. Fundamental distinction between these two groups. Monocots are things like corn and sugarcane and wheat. Dicots are things like spinach and soybeans and cabbage and stuff like that. These are typically take the form of a grass. They can oftentimes have multiple shoots, although corn only has one shoot. They're root systems tend to be dispersed. We call them fibrous or adventitious roots. Um, and once again, they have differences in how their vascular cambium, or their vascular tissues are arranged, but they're also different in terms of how their roots and stems are arranged. Dicots tend to have a long taproot with lateral roots coming off of it. As a result, they grow deeper into the ground. They're better anchored for also growing upright more. These things tend to have venation. The veins of the leaves tend to be parallel to one another. And so if you look at a grass blade, you can see the veins going through it, and the veins are all parallel to one another, as opposed to something like a maple leaf, where the veins go out and branch. They're not parallel to one another. So that's another difference. This is a dicot leaf. This is a monocot leaf. And you can see that in the venation here. You can't really see the venation here because they're all parallel. They're going in the direction of the leaf. These are all different dicot leaves. And as you can see, there's a single central vein and then a bunch of lateral veins going off of them. Lots of things that you could learn about leaf structure. Don't learn any of these things, but they're important for identifying different families of plants. The one thing that you should know is this one. This is a pinnately compound leaf. What that means is that there's a central stalk, a central petiole, and then there are leaflets, and they're paired coming off of it. And so pair, 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 and then a terminal leaflet at the end. And so the reason that that's important is that this group of plants that are soybeans, that are in a general classification of plant called legumes, they're in the family Fabaceae. These are the plants that are high in nitrogen. They have the ability to take nitrogen out of the atmosphere and fix nitrogen and put it in their bodies. And one of the reasons that we grow soybeans is because these plants are high in protein, higher in protein than corn is. Corn is a heavy carbohydrate containing crop. Soybean is a heavy protein containing crop. So if you're a vegetarian, you should be eating things like soybeans so that you're getting enough protein in your diet. So you should be eating diets that are high in legumes like soybeans and lentils and black beans and things like this, because that's a good source of protein. So this is a good protein source. One of the distinguishing characteristics of the family Fabaceae is they have pinnately compound leaves. So you guys know the locust tree here in Missouri? Honey locust, black locust. It's a tree that has these big spikes growing off of the trunks. If you look at their leaves, they have pinnately compound leaves, and they produce their seeds in these long pods. So any of your farm plants that produce pods, green beans, soybeans, lima beans, black beans that grow in pods that you have to open up the pod to get at the bean, those are all legumes. And if you look at the growing plant, they have pinnately compound leaves. If you're walking around campus right now, you will see trees with these long pods hanging off of them. When you look at the leaves, you'll see that they're pinnately compound. When you look at the trunk, you'll see that there are these big thorns sticking off of them. Those are locust trees, and they're in this same family. But other than that, you don't need to know anything else about this. Just know this one leaf form. All right, so this brings us to the structure of a leaf. So as these vascular tissues are going up the stem of the plant, they branch off and they continue into the leaf, into the petiole of the leaf and out into the leaf. And so you'll notice that xylem is on the inside and phloem is on the outside, so that when they bend and grow out into the leaf, xylem is on top and phloem is on the bottom. Okay? So when we look at a leaf, in cross-section, what we will see is xylem is on the top of the vascular bundle, phloem is on the bottom of the vascular bundle. And so this is the vascular bundle as it grows out into a leaf. And xylem was on the inside, phloem was on the outside. When it turned to go into the leaf, xylem's now on top, phloem is now on the bottom. So this leaf is basically a bunch of layers, kind of like a sandwich. And the bread on either side of the sandwich is the epidermis, this layer of cells at the top and the layer of cells at the bottom. You can distinguish them between upper epidermis and lower epidermis depending on whether it's on the top of the leaf or on the bottom of the leaf. That layer of cells kind of contains all of the other things that are in the leaf. And that includes the vascular bundle and these cells that are not part of the vascular bundle that are referred to as mesophyll cells. Mesophyll cells are where photosynthesis takes place. Okay? So once again, the only reason I'm going through the detailed structure of the leaf is because inside the leaf is where photosynthesis takes place, and inside only portions of the leaf are where photosynthesis takes place. That is inside the mesophyll cells. Well, what photosynthesis does is it takes carbon dioxide and water. It gets energy from the light. It uses chlorophyll to capture that light. Chlorophyll is a pigment. It's the green pigment that you have in leaves. And it uses an enzyme called rubisco, which we will talk about in a few minutes. And these things allow the energy and light to be captured by chlorophyll, and then rubisco is what eventually captures the carbon dioxide. And it's going to take that carbon dioxide and water, and it's going to convert it into glucose, which is the energy storage compound, the first energy storage compound in this process. And it produces as byproducts water, and another byproduct is oxygen. So the water the plant gets from the roots. The roots absorb the water, transport it through the xylem up to the leaves. So that's where it's getting the water. But the carbon dioxide that it gets is floating around in the air. And so when you have an upper epidermis and a lower epidermis that's kind of containing the internal layers of the leaf, there's no way for gases to get into the leaf. And so what plants have to do this is they have openings in their leaves. These openings are called stomata, plural, stoma is singular. And these stomata are basically holes in the leaf. And they're regulated by these cells on either side of the stomata called guard cells. And these guard cells, they either swell up or they shrink down depending on how much carbon dioxide is inside the leaf. So when they shrink down, the hole opens and carbon dioxide can flow into the leaf. Once the leaf has enough carbon dioxide inside of it, they close those openings up because at the same time that carbon dioxide is entering the leaf, water is evaporating out of the leaf. And so the plant wants to get enough carbon dioxide into the leaf so it can do photosynthesis while losing as little water as it can in the form of water vapor evaporating out of the leaf. And so rather than having a loosely organized epidermis, they have a very well-organized epidermis with these little pores in the epidermis that allows the flow of gas back and forth. The challenge for plants, the challenge for every plant is getting enough carbon dioxide in to do photosynthesis effectively while reducing the amount of water loss when those openings are open. This is the fundamental difference between C3 and C4 plants. This ability to open themselves up to gas exchange while limiting the amount of water. I erased it, but I told you that C3 plants grow best in what types of environments? Cool, moist environments. They tend to open their stomata up for longer periods of time, which allows for more carbon dioxide to enter rather, but it allows for more water to exit. So as a result of that, they don't do well in hot, dry environments because when they open up their stomata, while they get a lot of CO2 in, they lose a lot of water because it's hot and dry. So they tend to do better in cool, moist environments. C4 plants open up their stomata less often for shorter periods of time. As a result, they still get carbon dioxide in, but they reduce the amount of water loss, which makes them better suited for growing in hot, dry environments. And we'll explain specifically how they can do that, whereas C3 plants can't, when we delve a little closer into photosynthesis. But once again, you need to open yourself up to gas flow because it's these cells that are doing the photosynthesis, and there are air pockets between these mesophyll cells where air hangs out. But as the plant cells do photosynthesis, the air that is in those air spaces becomes less and less rich in carbon dioxide as that carbon dioxide gets used up to make glucose. Once that carbon dioxide falls below a certain level, the plant has to open up its stomata to get more carbon dioxide in there. Open up for carbon dioxide, lets water out. That can be a problem if you're in a place where you're water stressed. Does that make sense? Okay. Holy crap, that's the end. That sets you up for what we're going to do on Monday, which is talking about photosynthesis. So we're going to talk about, in general terms, how photosynthesis operates, but structurally, C4 plants have, around their vascular bundles, they have this extra layer of cells called the bundle sheath cells. And the bundle sheath cells are crucially important for what separates C3 photosynthesis from C4 photosynthesis. Yes, ma'am? Oh, crap, we don't have classes on Monday. We have class on Wednesday. Next time we meet, we'll go through this. There's a chapter, the next chapter in Plants and Society is about photosynthesis. It's already up. I just need to unhide it so you can go and read that. So as you read that, keep in mind the structure of the plant leaf as you go through and read that. Okay? All right, other questions? I keep forgetting. I tried to make an appointment with a student on Monday, and she's like, it's Labor Day. I'm like, oh, yeah, it's Labor Day. So, all right, have a good Labor Day weekend, and I will see you on Wednesday. Try not to drown on your way from one class to another today.